Hi, my name is Tessa, and I'm a 2025 ACD Fellow. I'm currently working on my PhD at Queen's University researching a novel gene replacement therapy for AGAT deficiency. Today, I will be extracting protein from mouse tissue. These samples are either from wild type, other known as normal mice, or mice that do not have the AGAT gene, so AGAT deficient mice. And then those AGAT deficient mice are either treated or untreated with a gene therapy. So here I'm using RIPA and protease buffer, which extracts the protein from the tissue and stops it from degrading. So I'm adding 1 ml to each tube, and then I use these forceps and extract the tissue from the previous tube into the special bead mill tube. I clean the forceps between every sample to stop any contamination between the samples there. So these tubes are special because they have these little metal beads at the bottom. They'll go in a bead mill, which I'll show you in a second, which then works to break down the tissue. So this is our bead mill here. It is basically like a big centrifuge, but it shakes all around instead of just on one plane. We have to make sure it is balanced, so we add an extra tube there, and then it does this orbital shaker here. So that means that all those beads in there are working to break up the tissue. So we take them out, discarding our balance, and then we grab all of our samples here. And now we put them on ice. So they're on ice for 10 minutes. This is to give the RIPA buffer time to extract the protein from the sample, and then the protease inhibitor works so that that protein doesn't degrade over time. So I labeled some extra tubes to put all the liquid from the bead mill tube into our new tubes. And then we are taking our tubes up to the 7th floor. So this is our big freight elevator where we go with samples, and we go to the sonicator. So this is just a quick video there. I forgot to actually videotape sonicating it, but sonicating is just another way of breaking up the tissue. Then we centrifuge for 10 minutes at 4 degrees at max speed. So this is like a centrifuge I was talking about before, so it just spins really fast. So we are putting all of our samples in there and then making sure it is balanced again, and it runs for 10 minutes, and that will bring any solid of the tissue that didn't get broken up, or fibers, protein, like things that are not proteins there, to the bottom. And I am just labeling 2 sets of tubes here, one for our main sample and another with a smaller amount for testing determining its protein concentration. So I'm just working here, taking out all of the sample. So I will show you in a moment here, that is the little pellet, as we call it. So all of the solid tissue that couldn't go down. And we want just the clear extracted protein. All right, now I'm doing another experiment to determine the protein concentration in those samples. So I make a standard curve of different concentrations all the way up from 2,000 micrograms to 0. So we make our standard there, and then we will add our samples. We dilute them so that they are within that working range. So I am just adding the RIPA, the same buffer that I showed you guys before, and then we add our sample in as well. So this is done in triplicates. So 3 little wells for each sample is what we do so that we can average them all out. And then we make what's called our working reagent. So this is fun because it's a little colorful. It makes this green color. And then when we pipette it onto our samples, however concentrated the sample is, the more purple it goes. So we put it in the oven there for 30 minutes to incubate, and then we go up to our plate reader and we make sure it's under the right wavelength and we read it, and then we have our little colors there. So then we're done with that plate, and this is just some quick analysis of that plate that I just did. So we determine our standards from here and their amount, and then we make our graph. We determine our squared and our intercept so we can figure out the concentration of our samples from there. So again, I am just going sample by sample to figure that out. We have to make sure that it is averaged first, the blank is subtracted, and then we can figure out our concentration. And then I just dilute the samples quickly from those numbers, and they go into the -20 until we need them again. Thanks.